Right. Any questions for the speakers? There's a question here.
Hi. Hello. Great talks. I have one for Dr. Novak. Yeah, so in terms of that variability that is seen across the patients in how in stimulation response, does that mean that in general there's a lot of variability in gene expression in the patients directly? Is that kind of benchmarked at all?
Yeah, so we do look at the gene expression profiles. There are similarities, there are differences. One thing that's really unique about how we do our analysis, we basically simulate the effect of the drug one by one on each patient and then see what the outcome is. So everything that I showed is the result of that. So it's many steps in that process, but it's the final analysis. Essentially, if you were to give this drug to a patient, what would happen? Would they do better? Would they do worse? Or just most drugs are just irrelevant. They just won't do anything. And so all the drugs that we found have some effect of the ones that I showed for a subset of the patients, right? Now, the study findings are, it doesn't have to be truly personalized for everyone. It's not that every patient is completely unique. There's nothing that could help all the patients, but there may not be one drug that could help everyone. And most likely there's these 2 or 3 groups that focusing on those just gives you so much more clarity about the underlying mechanisms, some of which align really beautifully with some of the ones we heard about this morning. But for any one patient, they may not hold true. And that's sort of the word of caution because every trial has non-responders. And so we essentially see that before the trial happens, right? And so one thing that we're doing is looking at who should we enroll in a given clinical trial based on likelihood of response. Because you want the trial to be successful. You also want to benefit patients and you don't want to have the adverse events. And so it's sort of a good alignment. And so it's not about the gene expression necessarily, and it's not at all only about Syngap. It's all the genes, but how they work in a network also matters.
We have a question over here. Yeah, I have a question about the milk exosome study, which is very interesting. Which splice isoform of Syngap have you packaged? And knowing that there are many splice isoforms of the protein, how do you envision delivering multiple isoforms so that they reconstitute physiologically appropriate stoichiometries?
Okay, good question. So what we did first of all was human Syngap, and I think it was the full-length Syngap, if this answers your question. But again, our PME technology, the big plan actually is to allow it for gene editing. So we really don't want to just deliver the gene, we want to edit or repair mutant genes, and we are funded currently by, well, it's not a whole lot of money, but we are funded by the new NIH Somatic Cell Genome Editing Consortium, and they want to see gene editing. They don't care about gene delivery. They really want to see gene repair, basically.
So a question for Richard. So you show that you have the RNA-seq data from Syngap patients, and seems like they don't the genotype is not so much predictive of the signature. But I wonder, as a group of the Syngap patients, can you extract some signature and use that to mine these data sets we have for all the other patients? You're able to find this more Syngap patients or the people carrying either non-coding variants or some not very obvious variants. Use that feature to find them.
Yeah, no, you're absolutely right. What's interesting is that it's not just about the mutation. It's all the other genes, all these SNPs, variants of unknown significance, everything, right? Also, the patients are on different medications. That matters. You can't ignore that. Now, to what extent it matters, who knows? But it does matter to some extent. And so that's how we look at our analysis. If you look at the gene expression, it might tell you something. Overall, and this is true for a lot of different disorders, genotype-phenotype correlation has not been very fruitful. There's a lot of cases where there are 2 different phenotypes that have identical mutations. Why? Who knows, right? What we're focused on is how do you fix it? I don't care how you got there, but let's fix it and let's figure out which patients would benefit. And so that's one of the kind of subgrouping analysis that I showed. by finding patients that are similar from this network standpoint, drug response standpoint, you absolutely can find these mechanisms. Now, one of the slides that got deleted in there was looking at, for example, at HDAC inhibitors. What's really exciting is that for any one patient, they're either a responder or very much a non-responder, and almost nothing in between. And it's like any of the HDAC inhibitors could work, or none. And so you could look at, a subset of those patients. If you're looking at the HDAC inhibitory mechanisms, maybe that subset actually would be very useful. And then you can look at, we're actually doing this for oncology, finding biomarker panels that can be predictive of a response. So just, like 5, 10, 20 genes, as opposed to the full transcriptome.
Yeah, can you comment on the size that you packaged into your milk exosomes, like compared to retroviruses and adenovirus, et cetera?
Yeah, great question. So the plasmid that we used is the smallest that we could find. I mean, bare bones, the size was about 7 kb. If we increase the size by using a slightly different plasmid, say 10,000 kb, the efficiency goes down, but it's still deliverable. So 10,000 is definitely an option. Right now we are playing with non-integrating vectors because that adds safety to the game. And I think they would be around 8,500 kb. So it's right in the range that we can deliver.
Yes. Well, you could use like that on your one top of the make sure it's all your cargo.
Can you say this again? Sorry.
My question is then, can you use another technology besides a prokaryotic plasmid-based, like for example, a doggy bone molecule or something that doesn't have the prokaryotic backbone to make sure you're maximizing the amount of genetic material you get into your exosomes? Have you thought about that?
We have not. But at the same time, we are pretty happy with what we got because we can show, we can deliver the gene. But good point.
As a follow-up to that question, sorry, how long lasting is this plasmid DNA that you deliver with exosomes? Are you modifying it with SETNA or some sort of modification to keep it long lasting?
So we currently in the process of doing dose response and time course studies. So I cannot really answer your question, but this is work ongoing in the lab right now.
Thank you.
I have a question. Oh, there's a question.
Yeah, hi. Mine's just a question regarding the PMEs. What would be your next sort of phase? You're delivering it directly now in the mice clinical phase. What would be your next step, I guess, in the research? Where would you start using, monkeys, or what would be the next phase in the delivering of these PMEs?
Yeah, good question. I mean, like I said, right now with our company, Staten and I, we're talking about, and my collaborator and I, we are talking about, probably non-human primates. That's one model that we want to do. We are not ready at this point to really jump into human studies. I mean, that's, as you know, is a very tedious and long-term process. And but non-human primates. We have done some work in large animals like pigs, and all the animals tested so far to date behave the same way as far as the pharmacokinetics of milk exosomes are concerned. So we are pretty confident that these pathways are conserved across all animal species. If this answers your question.
Do we still have time? No. Okay. Thank you. Well, I had one for each, so now I have to pick which one I want to ask. So actually, I'll ask you offline. So for with that large amount of data that you have for personalized, basically for each patient, were you able to find, and this is more from the genetics perspective, any gene expression pattern that could explain, say, the differences in disease severity? And I know that that's not the focus, but it's data that you could get from your analysis.
Yeah, we absolutely can. And so our goal is the therapeutic goal, but the idea is that this data is given to the foundation for research use, right? So others can certainly dive in, and from the respective hypotheses, you can absolutely do those analyses. And the metadata is there, so it's all, I mean, it's de-identified, but you have the drug information that for medications the patients are taking, and the controls, by the way, which is also very relevant for comparison. The genotype for Syngap and a whole host of other data. You absolutely can. What's interesting is that you'll start seeing some of these patterns. I mean, one thing that really struck me with the nitric oxide data is that overall it was decreasing in the patients, but for some patients it was actually going up. And so what I'm always excited about is the noise in the data. And that's essentially what we see here. There's no disease. It's a bunch of patients with a similar diagnosis, but how you treat them isn't necessarily the same, nor should it be. But absolutely something we could, the data is there to be analyzed.
Thank you. Let's thank the speakers one more time.